So when we talk about the cardiovascular system, cardio pertains to the heart and vascular pertains to the blood vessels. So I think I mentioned that we're going to use the discussion on blood and the the Zoom recording I made available to you on blood as a self-study segment. If I find we have some time to review it in the classroom, we will. But I think looking ahead at the schedule, it'll be better to keep the blood topic as a self-study topic, and that allows us to spend a bit more time on the heart and on the dynamics of blood vessels and so on. So for the first couple of things with respect to the cardiovascular system, just like any system, if we can kind of summarize that system's responsibilities, then anytime we get into some details and maybe get a little bit bogged down on what it is we're talking about, if we can refer back to what our summary statement was for the particular system, it can sometimes bring us back to where we need to be from an understanding point of view. So the thing to realize about the cardiovascular system is, first and foremost, it's a transportation system. And the goal of this transportation system is to deliver oxygen and nutrients like glucose and amino acids and so on to cells and remove or pick up carbon dioxide and waste from the cells. So the thing to realize is that's going to require some sort of a driver. So the heart drives particularly delivery, and venous return is a general term, but it's responsible for the removal steps. And the other part of this to recognize is that it's often described as a closed circulatory system, and arguably two of those. And we'll talk a little bit about why I say two of those as we move along. But we're going to recognize that we have what we describe as a systemic circulation, which is the one we typically think of, where the oxygen and nutrients are going to be delivered to all the cells in the body. But we also have what's called a pulmonary circulation, which we'll spend lots of time talking about, that is going to circulate to and from the lungs and has a distinctly different responsibility when it's going to and from the lungs. But again, we'll spend time talking about that and highlighting the differences when we get to those particular conversations. Okay, so let's consider some of the anatomical considerations. Today, we're going to spend our time talking about the heart. So the root word for heart is cardio. Anything to do with cardio typically has at least something to do with the heart, whether we're talking about cardiac muscle cells, whether we're talking about cardiac chambers or valves. If that word cardiac is associated with it, then it's going to be discussion on the heart. Another term that will be related to the heart is coronary. So we have cardio and we have coronary, which are indicators that we're having a discussion about the heart. So when we talk about the heart, first thing to realize about it, it is primarily a muscular organ, and it is described generally as a pump. In reality, it's two parallel pumps within one organ. One side or one of those pumps is going to pump to the lungs, and one of the other pump is going to pump systemically to all the other tissues in the body. So we'll see how there's some similarities and differences between those two pumps within the same organ. So from an anatomical point of view, the heart's about the size of a closed fist. And it's located in the inferior, what's called the mediastinum, which is the middle lower segment of the thoracic cavity. So if we drew in the thoracic cavity, which looks something like this, and a big bulk of the thoracic cavity is filled with lung, what we would notice is that the left lung has a distinct notch, and that notch fits or allows the position of the heart. So the heart sits in this inferior mediastinum between the two lungs, and the heart is oriented a little bit towards the left inferiorly. So the heart generally has what's referred to as an apex and a base. The apex is pointed downward towards the left hip, so the sort of angle of axis would look something like this, so that the apex of the heart, the so-called pointy end of the heart, points towards the left hip, and the base of the heart points towards the right shoulder. And we'll see a little bit later on that it's also slightly rotated so that the four chambers in the anatomical position don't appear to be equally distributed from an anterior posterior point of view. So I'll explain that when we look a little closer at the chambers of the heart and the way that it's arranged. So we get now where the heart is going to be located. And we can appreciate that the apex of the heart is going to be located, give or take, around the level of the fifth rib. So this may not be a completely anatomically accurate. And the upper part, the base part, is palpable just below about the level of the second rib. So probably in this diagram, all of this would be lifted a little bit higher within the rib cage. But that's going to give us our basic location for the position of the heart. The heart's covered by some connective tissues known as the pericardium, and it has an outer layer, is reasonably dense connective tissue that holds the heart in position. And it's sometimes referred to as the parietal pericardium. And then the inner layer, known as the visceral pericardium. visceral meaning organ, so closest to the organ itself. It's pretty much embedded. Whoops. Embedded into the next layer, which is the myo. So that tells you what tissue you can expect to find. Myocardium. And this visceral or inner layer of the pericardium is a sometimes called an epicardium as well. You might see it called an epicardium. Where am I going to put that? Also known as the epicardium. And it's right next to the myocardium and has fibrous connections into the myocardium that makes it pretty much very difficult to separate those two tissue layers from one another. And there's no reason to have them separated under normal, healthy conditions. So the idea between these two layers is that there's going to be the inner layer that allows the heart to contract and relax, and the outer layer that holds the heart in its location within this mediastinum with some fibrous connections to the base of the diaphragm and adhering to some of the structures associated with the thoracic cavity, like the sternum and so on. And then the inner layer beats with the contractile tissues of the heart, and there's a fluid between the two called the pericardial fluid, which is a lubricant to reduce friction between those two layers. So we understand that a heart beats at rest somewhere between maybe 60 and 80 or 90 beats per minute. And in order for that to occur efficiently, we would like the heart not to beat all around the chest wall, but stay in a relatively localized position. And that's the role of the outer layer, while the inner layer beats with the contractile pattern of the myocardium, and the lubricant in between allows those two functions to occur with minimal resistance. So hopefully that makes sense to everybody as far as that goes. We okay with this so far?
Yes.
Yes.
Okay. So this discussion we've just had really represents the outermost wall of the heart. Okay. Now, when we go to the next layer of the heart, the middle layer, is the myo. So what tissue do we know is going to be associated with the myocardium?
Yeah, muscle cell.
Myo gives you a root for what? Nobody can tell me what the root myo, M-Y-O, indicates?
Muscle?
Muscle.
Yeah, muscle. It wasn't supposed to be a hard question. Good. Somebody was brave. So myo is muscle. In this particular case, we're talking about cardiac muscle cells or tissue in the case of the layer. And when you took AMP 100 and you talked about muscular tissue, you should have had a discussion about skeletal muscle characteristics, cardiac muscle characteristics, and smooth muscle characteristics. And one of the things that should have been mentioned then that is assumed to be understood now is that cardiac muscle cells have characteristics of skeletal muscle cells and characteristics of smooth muscle cells, But what makes it unique is where it's found, which is here in the myocardium. So we understand that the myocardium is the innermost layer, okay? And I'm going to mention, because otherwise I'll forget, that the innermost layer is called the endocardium. And the endocardium is mostly an inner lining of the chambers of the heart, continuous with the blood vessel linings that enter and leave the heart. And we'll talk a little bit about what those are made from when we do the blood vessels. But the idea is that this endothelium, innermost lining of the blood vessels and heart walls, is designed to be very, very smooth in order to minimize turbulence or disruptions in blood flow. So we want a nice, smooth inner lining that allows the blood to move without being disturbed. If you disturb blood or agitate blood, sometimes you initiate blood clotting mechanisms, and we don't really want that to happen inside the heart. So that endocardium is going to be made up of this lining, often of simple squamous or sometimes stratified squamous epithelia, creating a very smooth surface. What we do want to spend a bit of time on is some of these characteristics associated with the myocardium. So remember, we said briefly already that the heart acts as a pump. In fact, two parallel pumps within one organ. So if we're going to act like a pump, that means we're going to have the capacity to contract. And that's going to be the responsibility of this middle layer, the myocardium. And the myocardium is made up primarily of cardiac muscle cells, also known as cardiomyocytes. That's saying the same thing two different ways. So a couple of things about these cardiac muscle cells that makes them very unique. And when we have a tissue or a cell that's very unique, especially if we spend a bit of time talking about those unique features, it's probably worth your while to make sure you review this next segment and make sure you're pretty comfortable with it. So we have some similarities to skeletal muscle because these cells are striated, and hopefully you remember that the striations that were visible in skeletal muscle were created by dark bands and light bands, the dark bands consisting mostly of myosin, and the light bands consisting mostly of the actin, which usually was associated with tropomyosin and troponin. The same concept applies with cardiac muscle cells. The arrangements are not quite as organized, but the striations do appear. So cardiac muscles look striped, but they are typically uninucleate, which is more similar to smooth muscle cells, whereas skeletal muscle cells were multinucleated. You can get the odd cardiac muscle cell that might have a second or even a third nucleus, but most typically they're described as being uninucleate. Now, another feature that makes them unique is that they tend to be branched. They branch quite freely. So we might have a cell body with a series of branches, and here's our nucleus in the center. So where it branches would associate with another cardiac muscle cell. So even a cardiac muscle cell that would communicate this way with another branch over here, and this might be a separate So the way I've drawn this is we've got we've actually got 4 distinct cardiac muscle cells here. 1, 2, 3, and 4. So again, quite different than the way that the skeletal muscle cells were arranged. Muscle cells in the skeletal muscle tissue are arranged in bundles. They didn't branch, so they just bundled up side by side. And in the cardiac muscle tissue, we're going to have these branches that create sort of more sheets of muscle tissue, which again is somewhat similar to smooth muscle. But again, we're talking about cardiac muscle cells that have characteristics from both smooth muscle and skeletal muscle features. So these cells require a lot of ATP. They make a lot of ATP. So they're going to have lots of mitochondria. And this is typical of most muscular cells. They all have to be able to contract. In order to contract, they need ATP. In order to relax, we need ATP. So there needs to be a lot of ATP. And therefore, we're going to see lots of mitochondria. We see a complex compound called myoglobin. Myoglobin sounds a little bit like hemoglobin, and it does a similar thing to hemoglobin. It carries or stores, if you like, oxygen. So cardiac muscle cells, just like skeletal muscle cells, can store some of their own oxygen so that in scenarios where there may be a reduced blood supply to that cell, it has some stored oxygen in order to sort of withstand that time that they may be have a reduced blood supply for. So myoglobin and glycogen. And who can tell me what glycogen is?
It's a form of glucose.
Well, kind of. What kind of form of glucose? So the person that's speaking is being very brave. Okay, let me just start by saying that. The person that's speaking is at least trying to create a bit of a discussion, probably because they recognize how tortured this presentation is without a little bit of discussion. So kudos to that person for making an attempt. What's very disappointing is that this isn't common knowledge by now in the classroom. There isn't a single student listening right now that should not be able to answer what glycogen is.
Is it a
And if you're
stored
unable
form
to,
of
Is it a
glucose?
form of carbohydrate?
well, again,
Carbohydrate?
no, it is, but you're showing me that you don't really know what you're talking about when you make comments like it's a type of glucose or it's a type of carbohydrate. You need to be much more specific. What are we having a discussion about here? Mitochondria. What do mitochondria
Energy.
produce?
Source
ATP.
of energy.
No, that's not the answer. Listen to me. Let me finish now. Okay? What do mitochondria do? They make ATP in the presence of 2 primary products, oxygen and glucose. I mentioned that the cardiac muscle cell can store oxygen in a molecule called myoglobin, which means that there is some ability to survive a period of time without a blood supply. Now, the other thing that the blood supply is responsible for that we started today talking about is the delivery of nutrients. In the case of the mitochondria, the favorite nutrient is glucose. This cell can store glucose as glycogen. What is glycogen? Glycogen is a molecule of glucose plus glucose plus glucose plus glucose to make it a polysaccharide. It is stored glucose that can be easily broken down into its building products, glucose, to be made available to the mitochondria for ATP. So these two words that I've just written, myoglobin and glycogen, are about availability of the easy products required to make lots of ATP. If we have a good blood supply, we get the oxygen from the blood supply, and we get the nutrients like glucose from the blood supply. In scenarios where the blood supply is not good, we can take advantage of this myoglobin and this glycogen. Okay, so it's a very good specialty feature for these particular cells. Now, skeletal muscle cells store glucose as glycogen and oxygen as myoglobin as well. So this is not unique to the cardiac muscle cell, but it is still a pretty effective backup plan for a cell that has to make a lot of ATP. So what's going to make a cardiac muscle cell unique is a couple of other features that we should be aware of. One of those is that cardiac muscle cells have a property called autorhythmicity. And what that means is if you took them out of the body and put them into a dish with some glucose and some oxygen and some other nutrients into a nice, comfortable environment, they would beat independently all on their own. So autorhythmicity is autocontraction is really what we're saying. These cells have the capacity to contract independent of an electrical signal. You remember that skeletal muscle needed an electrical signal from a neuron in order to depolarize. These cells have the capacity to beat in the absence of an electrical signal from a neuron. But they beat in a type of performance that's referred to as autorhythmicity, and it is independent of other cells. So what we would notice is that in one part of the heart wall, for example, up in this area, the cardiac muscle cells up in this area will beat much faster than they do down in this area. So we'll see a little bit later on today that we're going to require a system that coordinates this feature of autorhythmicity. So it's pretty unique, and it's going to require some coordination, which we'll find out about a little bit later. Now, the other thing is that these cells connect to one another at some very unique and specialized connections. And those connections are referred to as intercalated discs. And they're formed by desmosomes. And desmosomes are protein extensions from one cell that are going to connect with protein connections of the adjacent cell to form a nice tight lock, so that when these cells contract, they won't be separated from one another. And by gap junctions. So that if we looked closer at an intercalated disc, let's go to another page here. So we're talking about here's cell 1, and our cell 2 might be kind of locked a little bit together like that. And these desmosomes would lock up in different segments to make sure that when these cells begin contracting, they won't be pulled apart. And between those desmosomes, we're going to see some gap junctions. So these represent some gap junctions. Now, hopefully you remember when you had a discussion about skeletal muscle cells, you remember that a skeletal muscle cell had a resting membrane potential, which was more negative inside the cell than outside. And when we depolarized the skeletal muscle cell to a threshold, it caused a cell-wide depolarization. It was what was called an all or nothing response. Get enough of a positive charge inside the cell to a threshold, and then the entire cell would depolarize. Do you remember that? Does that sound familiar?
It does.
Yes.
Okay, so the same concept's going to apply for this cardiac muscle cell, but the cardiac muscle cell can depolarize independent of other cells and other signals, as well as from electrical signals. And we'll talk about what some of those are. But the point is, the concept's the same, a resting membrane potential that's more negative inside than out. And for example, if sodium cells rush in, or calcium cells, ions, I should say, rush in, and they depolarize to threshold, the advantage of these gap junctions is that the ions can pass through to the next cell and cause a much faster depolarization than if we had to wait for another signal from a neuron. So these intercalated discs create tight junctions with easy transfer of ions from one cell to the next. So this is going to allow large numbers of cells to function as a unit. Okay, so that's also described as a functional syncytium. So remember, anatomy and physiology was about structure and function. Here's an example where we're using the word function and this other word, syncytium, that maybe some of us have never heard of before. But a syncytium is describing a group of cells, in this case, that are functioning as a unit. Okay, now we describe this one as a functional unit because each cell is still an individual cell, but because of these intercalated discs are connected to one another, and the fast transmission of those electrolytes allows cells to contract in a sequence as a unit. Think of it as a wave of contraction. And we're going to talk about the importance of that a little bit later on today. It's extremely important. So a functional syncytium is as opposed to a structural syncytium. So you may not be aware of it, but you've already talked in A&P 100 about a structural syncytium. So the best example, because of what we're talking about right now, of a structural syncytium is a skeletal muscle cell. You remember that a skeletal muscle cell was a long cell that was striated with light and dark banding all the way along its length and pushed out to the outside all the way along its length were nuclei. The skeletal muscle cell is described as a multinucleated cell. But embryologically, it was a whole bunch of individual cells. So during the embryological development of a skeletal muscle cell, maybe 50, 60, 70, as many as 100 individual cells blended together to create one large cell. That would be describing a structural, because now it's all a single unit. That would be described as a structural syncytium. Functional is telling us that the cells are still separate from one another. Structural tells us that they've blended to create a single one. Are we okay with that difference?
Yeah.
Yeah. Okay. So the other thing is, when we look at some differences between this cardiac muscle tissue and skeletal muscle tissue, is that when we generated an action potential in skeletal muscle tissue, we had positive ions leak in until we reached a threshold. So we said resting membrane potential was maybe around -70. If we got to a threshold of about -55, we would get this all or nothing response, and it would zoom up to maybe as much as a +30 before it had to repolarize and reset to get ready for the next contraction. Right? Is that familiar when you're looking at the pattern of an action potential in skeletal muscle?
Yes.
Okay.
Yeah.
So in cardiac muscle, we're going to start, and the primary electrolyte that was responsible for all of this was sodium, right? In the cardiac muscle cell, the early steps of depolarization are almost identical. We have a resting membrane that's pretty close, and for our purposes, pretty close to get to threshold. And we're going to see the initial rise up to about +30. So all of that's exactly the same. But here's where it gets different. When we reach that peak at +30, we're going to see a plateau before we begin the repolarization process. And as a result, this plateau causes a longer contraction phase. So this early steep curve is the responsibility for sodium, just like it was in skeletal muscle. But then some channels open up for calcium. So this is coming later and lasting longer. These calcium channels are described as being slow, and the sodium channels are fast. But the important thing to recognize is that we created a plateau. So the action potential and the length of time for a contraction to occur in the cardiac muscle is going to be extended. This is cardiac, and this is skeletal. So what that ends up looking like in our contraction phase is that for our skeletal muscle, the time for a contraction is going to sort of mimic the action potential. So we had a very fast action potential that was short-lived, and we went back down to base pretty quick. And our contraction cycle will kind of give us a little bit of the same sort of thing. That's what our contraction would look like. In cardiac muscle, because of the plateau, the contraction time is going to last longer. So cardiac muscle has a longer contraction phase to it, and that becomes important in the functioning of cardiac muscle tissue. When we want to beat the heart in a cycle, this longer contraction time is helpful. It's going to help create a stronger pump. Okay, there's some more details to what that gives us, but for our purposes, we just want to understand that this second flow channel for calcium produces a plateau that gives us a longer contraction time, which is an important part of cardiac function. So, and the reason I like to spend a little bit of time on this at this point in our talk about the heart is it helps us to understand why blood levels for sodium and calcium are so important, why homeostasis of sodium and calcium is so important. If we don't have adequate amounts of sodium, we can't get contraction of any muscle cell effectively. And if we don't have calcium in addition, we're not going to get proper cardiac function. Now, you remember, I hope you remember, that calcium in skeletal muscle is stored in the sarcoplasmic reticulum. You remember that from skeletal muscle?
Yes.
Okay. So when the cell depolarizes, the sarcoplasmic reticulum releases calcium. Remember, calcium would then bind to troponin. Troponin changed shape and made the binding sites on actin available to myosin so that we could get a power stroke, right? First, we got a cross bridge, then we got a power stroke. So all of that is very similar in cardiac muscle, but the sarcoplasmic reticulum it doesn't store quite as much calcium in the cardiac muscle cell as it does in the skeletal muscle cell. So the calcium that comes in this pathway ends up having a second role to play. It also binds to troponin. In addition to the calcium from the sarcoplasmic reticulum. So that phenomenon, that calcium that comes in through channels to the heart actually stimulates the release of calcium from the sarcoplasmic reticulum, is called a calcium-inducing calcium release. So it's a little bit of a double step or a double a location for calcium that comes from two places. The calcium that leaked in from the slow channels that causes the troponin to be released from the sarcoplasmic reticulum. So it was induced to release calcium from calcium. So if you do a little bit of reading and back up on cardiac muscle contraction, you might see that phrase, calcium-induced calcium release, and that's what it's referring to. The role that this calcium that came in from the leaky channels, slow leaky channels, established a plateau, but also stimulates the sarcoplasmic reticulum to release calcium, just like we saw in skeletal muscle, but the mechanism's a little bit different. Okay, so all of this little conversation is designed to show you some of the similarities and some of the differences between skeletal muscle and cardiac muscle. Okay, so what you want to get from this at this point is fast sodium, slow calcium gives us a plateau for a longer contraction cycle. Autorhythmicity is something we're going to have to deal with a little bit later on to help us understand how we're going to coordinate muscle contraction in this parallel pump. Okay, so all of this is a discussion about that middle layer, the myocardium. Is everybody with me still?
Yes, sir.
Okay, so let's take a look. Not that picture, we've seen that picture already. So here's what our heart looks like in if we pull it out of the chest from that sort of mediastinum area. And we can see that the bulk of what we're looking at, I'll outline the whole heart in the black, okay? That outline I just created in black is giving us the heart proper. And within the heart proper, we have those three distinct layers: the pericardium, the myocardium, and the endocardium. What we're looking at when we look at the outside of the heart is the pericardium. And the yellowish stuff represents adipose tissue. It's going to help insulate the heart a little bit of protection, but it also provides a source of fatty acids that the heart can use if glucose isn't readily available for the production of ATP. Okay, now, this chamber up here and over here represent atria. There's two of them, so that's a plural. If we were looking at a single one, it would be an atrium. And the two lower chambers are called ventricles. Now, the ventricles themselves, they're the real pumps, okay? They're the ones that are going to pump. And the atria are described as receiving chambers. So when we start to draw out the pathways of blood flowing through the heart, they're going to flow through these chambers in a one-way fashion. So we have a total of 4 chambers, and we have 4 valves. 2 sets. One set is known as the atrioventricular valves between the atrium and the ventricle, and the other set are known as the semilunar valves. So if we looked at this heart and we split it up into 4 chambers, if we did a cross section through this heart, it would basically look something like this. And then you can see that the orientation is off to one side a little bit. I'm going to even it out a little. So we're going to divide the heart into a right side and a left side. Remember, the atrium was the upper chamber. So we've got a right one and we've got a left one. And I'm drawing atrioventricular valves. And these lower chambers are the ventricles. And the ventricles are the real pumps. Now there's a myocardium that's part of all of these chambers. All of these chambers have the ability to contract, and that becomes an important part of delivering blood. Okay, but the chambers down below the ventricles are strong chambers. They're responsible for, let's see if I did this right. Okay, and this left side has got an even thicker. Now let's see if I did that right. So those red layers now represent the myocardium, okay? And our inner layer is going to be described, and it doesn't show up very well. Let's make it in green. So this inner layer now is our endocardium. Okay, inner layer. So the black lines represent the pericardium, the red represents the myocardium, and the green is our endocardium. So our valves are going to be extensions of the endocardium, the innermost lining. So the other thing to realize is that if the atria are receiving chambers, the right atrium is going to receive blood that is drained from the upper half of the body and the lower half of the body. So blood is going to return to the heart and enter the heart through the right atrium. From the right atrium, it's going to be delivered into the right ventricle. Now, the semilunar valve is up in this upper central portion of the ventricle. So the blood ultimately is going to be delivered out of this valve, in through this valve and out through that valve. And that valve is going to leave the heart through a trunk that goes to the right and to the left lungs. So if we followed our blood pathway, we're going to leave the right side of the heart and go towards the lungs. So this structure that we're looking at that's in purple on this picture right here, the number 2, is called the pulmonary trunk. And it divides into a right and a left pulmonary artery. Okay, and this one would be the right pulmonary artery, going to their respective right and left lungs. That's where that terminology is coming from. So the trunk is before the division. Okay, yeah, everybody's with me so far?
Yes.
Okay,
Yes,
and I've
sir.
designated the blood vessels, whoops, that carry deoxygenated blood in the color blue. So that's what we see here. These are veins carrying deoxygenated blood. This big one up here is called the superior vena cava. That makes this one here the inferior vena cava. So they're draining the blood from all over the body back to the right side of the heart. The blood is going to enter, in other words, be received on the right side, right atrium, and then delivered to the right ventricle, and the right ventricle is going to pump up to the lungs. So that would be referred to then as the pulmonary circulation. When the blood comes back to the heart, it's going to come back from the lungs from both sides. Okay, so this now starts to get a little bit crowded, but it's going to come back from the right lung and the left lung and enter the left atrium. And from the left atrium, it's going to enter the left ventricle. And from the left ventricle, it's going to leave in the left semilunar valve, which it means it's going to leave through a very large vessel. Most of you have heard of this vessel. Actually, let's take that out of there. We're going to just tuck in behind here. Anybody know what this vessel is going to be called?
Aorta?
That's right, the aorta. So it's the large blood vessel that leaves the left side of the heart carrying oxygenated blood to the cells and tissues of the body. So that becomes the systemic circulation. So I've just super simplified it to make it one big exchange location, but this would represent all the cells and tissues in the whole body, including the brain above the heart and the big toe below the heart. You follow me on that? So that the red vessels carry oxygenated blood and the blue vessels carry deoxygenated blood. Okay, so here's our aorta. Let me put it in another color. This represents our aorta here with some of the branches that we're not going to worry about naming, especially today. Okay, this blue structure here was the veins returning deoxygenated blood back to the right side of the heart. We delivered through the purple vessel to the lungs and then came back through vessels that we can't see here. They would be coming back to the left atrium, left ventricle, out through the aorta for delivery. The idea is 4 chambers and 4 sets of valves that are designed to make sure that we create one-way flow. In effect, we're going to create 2 circulatory pathways. The right pump pumps to the lungs. The left pump pumps to the whole body. Because the left pump has to pump to the whole body, it has to overcome much more resistance. So we see that in the thickness of the contractile segment, the myocardium, in the left ventricle is much thicker than it is in the right. Does that make sense?
Yes, sir.
Yeah, we're okay with that?
Yeah.
Okay, think about where the lungs are. Remember from our very first picture, we drew the heart right between the lungs. So the distance is close, and even from a gravitational point of view, not much work against gravity. So the right ventricle has to overcome very little pressure compared to what the left one does in order to pump the systemic circulation. So this is essentially what we're trying to illustrate here in this picture. Here's a picture of our muscle cell and the way that it's arranged in sheets. So this represents our intercalated disc. If we looked at the intercalated disc, we've got our desmosomes that create connections, and then we've got gap junctions that allow for easy transportation of electrolytes from one cell to the next. We can see the branches here, so we get some of the physical features from this picture, and we understand what some of the biochemical features are with respect to sodium and potassium or sodium and calcium for contraction dynamics and that kind of thing that we've already spent some time talking about. So let's take a look at this same thing, because understanding this idea that we're going to have 2 circulations, 2 pumps responsible for the 2 circulations, and I'm going to try to color code them for you a little bit. So we're going to divide our heart into a blue side and a red side. Okay, and on the red side, we're saying that the blood that is going to travel through the chambers on the red side is oxygenated. So it's got its full complement of available oxygen. And on the blue side, it's not that it has no oxygen, but it has less oxygen. Okay, So if we go down here and look at some cells that represent cells from the body, these could be muscle cells in the big toe, in the calf, could be brain neurons from the central nervous system, but they represent systemic tissues. Okay, so when we deliver, when we deliver from the aorta, we branch and branch into progressively smaller blood vessels until we get into capillaries. And capillaries are going to be little networks of exchange vessels that form in and around all the variety of cells that require oxygen and nutrients like glucose for delivery. So what we're drawing in here is our systemic delivery to the system. So we'll just once we've offloaded some oxygen, the arteries become veins, and we're going to leave the location of exchange, and we're going to return back to the heart. So this describes our closed circulatory system, closed in reference to the external environment. There's no direct exposure to the external environment. Now we've returned blood to the right atrium. Okay, so this is our atrium and this is our ventricle. And the blood is going to enter the right atrium, ultimately enter the right ventricle through the AV valve and leave through the semilunar valve. And when it leaves the semilunar valve, it's going to leave through a pulmonary trunk that divides into a right and a left pulmonary arteries that go to our right lung and our left lung. So we've begun our pulmonary circulation. This is systemic. And up here, it's going to be the pulmonary. So we can see with the systemic circulation that it was all about pumping blood from the left ventricle through the aorta for delivery to all the cells and tissues in the body. So we're going in that direction. Then we're going to pump, or we're going to deliver blood back to the heart on the right side, and it's going to be delivered to the lungs. The lungs, remember, are going, well, you might not remember, but the lungs are receiving air from the atmosphere so that we can have new oxygen available in the lungs all the time. Okay, and what that means is that the lungs then are going to send oxygenated blood that got picked up there. I'm just going to put it in behind here. I'm not going to try not to make it too complicated. But in this case, it's coming back from the two sides and then enters the ventricle to then begin a new circulation. So in our pulmonary circulation, we came up, we went right, we went left, we came back, we came back. That's our pulmonary circulation. Do you see that okay?
Yeah.
Yes, sir. It's clear.
Yep. Okay, and then our systemic circulation, can we do it with a different color that will show up? we'll do it with our systemic circulation is going to leave through this valve, the aorta, and to all the cells and tissues, and then back up to the right side of the heart. Leaves the left side, comes back to the right side. In the case of the pulmonary circulation, it left the right side, came back to the left side. It left as blue, came back as red. And that indicates, or what I hope it indicates, is we got our oxygen from breathing it. The missing oxygen is picked up, and now our blood becomes oxygenated to the left side to be pumped to all the body. Right? So right pump goes to the lungs to pick up more oxygen and unload CO2. A left-sided pump goes to all the cells to deliver O2 and remove CO2. So this is describing the flow of blood, one way through the right side of the heart to go to the lungs, and one way through the left side of the heart to go to the entire system. This aorta will branch and go upward to the head and neck and downward to all the rest of the parts of the body. So I've just simplified it by not including all those branches, but hopefully we recognize that this represents cells and tissues from all over the body to become our systemic circulation. Does that make sense? We're okay with that?
Yes, sir.
Yeah.
Yeah.
Or you just, yeah, you got it. Okay. So we understand that the right side's responsible for the right side's responsible for pulmonary circulation, the left side's responsible for systemic circulation. Now let's look a little closer at how blood flows through these chambers. So we'll draw the heart as an entire unit. Base, apex. Okay, and what we can even do here is include our layers. So our endocardium will be our innermost layer. And it will form our valves. We're going to start to put some names to this stuff now. Okay, so same thing, a little tedious, I know. So the green lines represent our endocardium. The myocardium is represented by the red lines. And we'll fill that in. There's our myocardium. Okay, remember we said that the myocardium of the left wall will be thicker than the myocardium of the right wall because of the increased resistance that it has to be able to pump against. So I'm not going to put that in there today just because it starts to get complicated. So this is going to represent the semilunar valves. What we want to recognize about our 4 chambers and our 4 valves is that they're important for ensuring one-way flow. So in other words, the AV valve on the right side, it's also known as the tricuspid valve. So our AV valves, we'll talk a little bit about them. The right one has 3 cusps. So it's a tricuspid valve. And the left one only has 2. So it's a bicuspid valve, also known as the mitral valve. I don't like that name because it doesn't give us any information. That word mitral doesn't tell us whether it's on the left or the right or anything structurally about it, how many flaps it might have. At least if it's called a bicuspid valve, we know that it's got 2 flaps, the bi telling us 2. Okay, so remember we said atria were receiving chambers. So the atria are going to receive blood from the inferior vena cava and the superior vena cava. So the blood is going to enter systemically from everything above the heart through the superior vena cava and everything below the heart through the inferior vena cava. And as it fills up the atrium, there will be more pressure in the atrium so that these flaps will fall open. Okay, so these flaps have little tendons attached to them. And when the valves are open, these little tendons are slack. So I've drawn them as ziggies. And they're connected to the ventricular wall. So they're going to be connected to the wall. So they're anchored. And these flaps that help to hold the valves open or closed are called the chordae tendineae. Now just okay. So where were we? We're talking about the chordae tendineae. The chordae, so that's telling you there's chords, and plural, there's more than one, and they're kind of like tendons. So that's all like that, and they're attached to the AV valve flaps. When the blood is flowing in, the pressure is higher in the atrium than it is in the ventricle. So these flaps flop open. That's just passive. And then the blood starts to fill up inside the ventricle and eventually starts to fill up so that we're going to create some pressure in the ventricle. And if the pressure in the ventricle gets high enough, it causes those valves to slam shut. So when the valves slam shut, they're going to look a little bit like this. They're going to be shut, so they kind of flop over, and these wrinkly tendons now are going to get stretched tight. And when they get stretched tight, that's going to prevent the flaps from over closing, from flopping inward. Does that make sense?
Yeah.
So they're attached to the AV flaps and the ventricular wall. When pressure in the ventricle forces the valves closed, the chordae, tendonae prevent inversion. Okay, so here they are flopped open, and when they are slammed shut, the chordae tendonae, they're attached to the ventricle wall on this side, so they get stretched, but they can only stretch to a point where they get tight. Once they're tight, the valves can't keep folding inward, so that's referred to as inversion. These chordae tendonae prevent inversion, and that's going to become important because we don't want backflow. So this prevents backflow. So to finish our thought here, and prevents backflow. So this is the purpose of these chordae tendonae on both sides. We're going to find out in just a few minutes that when the right pump works, the left pump's pretty much working at the same time. So as we fill blood up on the left side, it's going to be oxygenated blood, whereas on the right side, it's deoxygenated blood. But the idea is that as blood flows back to the heart after being delivered to the lungs, it's filling up in the atrium and then being delivered to the ventricles. And as the ventricle fills up, eventually the pressure in the ventricle is going to get greater than it is in the atrium, and that will force the valves on the left side to close the same way they already closed on the right-hand side, and the chordae tendonae play the same role on this side as they did on the other. So to move blood through chambers, we need one-way valves and we need pressure changes. That's the other thing that we need to talk about. Pressure change is going to come from stretch on the chamber wall, just like blowing up a balloon, right? The more air you put in a balloon, the more pressure you put on the wall, the greater the pressure inside that balloon. If you just put a little bit of air in a balloon and let it go, it just drops to the floor. If you put a lot of air in a balloon and let it go, it sputters and flutters all over the room before it drops to the floor. So that's describing a feature that we're going to talk about in the heart called preload. We're putting a stretch on the wall, in this case of a ventricle, that's going to give us ultimately a more forceful contraction, but it's going to help increase pressure. Right? The other feature that's going to increase pressure is when the muscles themselves contract. That's going to reduce the volume for the blood, and that's going to create a big increase in pressure. And what we want to see happen is that when we get enough of an increase in pressure, it's going to open up these two valves. Those two valves, remember, we called those the semilunar valves. So the right semilunar valve is from the right ventricle to the pulmonary trunk. And remember, the pulmonary trunk was going to branch into right and left pulmonary arteries to go to the lungs. The left semilunar valve is the valve that is between the left ventricle and the aorta. and that's going to ultimately result in delivery to all the cells and tissues of the body. So what you'll notice is that when we close one valve, so we closed this valve right here, and that allowed us to open this valve. When we close this valve, no more blood can go backward, and the only place the blood can go is out that semilunar valve. Same concept. If we close up this valve and open up that valve, the blood has only one place to go. Okay, and those valves will pop open when the pressure in the ventricle is high, and they'll drop closed when pressure is low. And we'll talk about all the factors that can change those pressures in as we move along in our discussion today. Okay, but what we're going to do now is take a short break so that we can take a breath, we can get some sustenance, we can get up and exercise a little bit, that sort of thing, so that we can be fresh when we come back. The way the heart sits in the mediastinum is rotated slightly such that we see most of the right-sided chambers. So we'll kind of depict that this way and divide the atrium in that kind of way. So in the reality, when you look on the outside of the heart, we're not going to see these divisions, but this is just going to give us a viewpoint that we want to be aware of. So coming off of the left ventricle is the aorta. So it would kind of leave the heart this way, and we would just clean up this mess a little bit like this. Okay, so that represents the aorta, and the very first branches off the aorta are going to be what are called the right and left coronary arteries. First branches off the aorta equal the right and the left coronary arteries. So the terminology can be a little tricky. Arteries we think of as being the vessels that carry oxygenated blood, but that's not always the truth. Now, we're okay with things not always being the truth because we understand that we never say never and we never say always, except always remember to say never or to never say never or always. So there's exceptions to the rule. That's what all of that mumbo jumbo really means. So the one consistency, though, is that any blood vessel that leaves the heart, goes away from the heart, is described as an artery, no matter what kind of blood it may carry. And if it is entering the heart or going towards the heart, in other words, the blood flowing in the vessel is going towards the heart, then it's going to be called a vein. Okay, so these are called aorta. The aorta and the first branches off are called arteries because they carry blood away. It so happens they fit the convention that they're carrying oxygenated blood, and in this case, they're carrying oxygenated blood to the heart. That's why they're called coronary arteries. So the left coronary artery is going to come off and divide into a branch that goes around to the back side of the heart. So it becomes the circumflex artery. So it's coming off of our left coronary artery, and it's going to have a branch that's called the circumflex, and it's going to have another branch that's called the anterior interventricular artery. And sometimes you'll see the word descending in there. So that second branch would travel down between the two ventricles. So it literally is going to travel down this border right here. So you can see left divides into two separate branches. The right coronary is going to divide into a marginal artery. So it's going to divide up and it's going to go right down the edge here, which is where that word marginal comes from. It's kind of going down the margin of the border. Okay, and then the second branch is the posterior interventricular artery. And also often we'll have that word descending. And that's just giving you the direction. It's going downward. So it would branch off like this. It comes around behind. And because we would be on the back side, I'll just draw it in a dashed kind of line. So it's on the back. We can't see it in this view, but it's between the two ventricles. And because the heart is rotated, these interventricular arteries are offset from one another. We see the anterior one and the descending one are offset from one another. On the backside, you can see most of the backside would be left ventricle, and most of the front side is right ventricle. We okay with that? We see that okay?
Yes, sir.
Yes.
Okay, so here's our one branch coming down like that. And the other branch is going around on the back side. So back side. And this branch is off anterior. So the idea, what I want you to get from this is that right now, how many different arteries are supplying the entire heart wall in this heart? So I'm talking about the branches now. How many branches of those first two arteries supply the entire heart wall?
4.
Four.
Right, 4. 1, 2, 3, 4. Now, if you've known somebody who's possibly had bypass surgery, you may have heard of someone who's had a quadruple bypass. The quad refers to these 4 arteries. So you can appreciate that if let me clean this up a little bit for the purposes of this. I'm going to get rid of the artery that's at the back because it just takes up space and sort of confuses us a little bit. So you can appreciate that the anterior interventricular artery, this one we can see here, is going to supply the anterior surface of the heart wall, both ventricles. So you can appreciate that if there was a blockage, whoops, I don't want that blockage. If there was a blockage right here, then this entire portion of the heart is going to be without a blood supply. So that would otherwise be called a heart attack. And this has affected a very large portion of the heart wall. There's a good chance that this person is not going to survive. It's affecting too much of the heart wall. The cardiac muscle cells can't get enough oxygen or glucose in order to continue to contract. This part of the heart wall is going to die and be replaced by scar tissue. You can't get enough pumping to go. So what happens hopefully, before this part is that this partially breaks, this partially blocks. So you get a partial block. And the person notices the partial block because they get some chest pain. They get it investigated, determine that this anterior interventricular artery is getting blocked, and they go through a surgical procedure that's designed to bypass that location. So instead now, we create a bypass. Okay. So that would be a single bypass. You can have a double, a triple, or a quadruple bypass, depending on whether it's 1, 2, 3, or 4 of these major arteries that need to be bypassed. So the younger your heart, the more typical this is the arrangement. The older that your heart gets, then the more what we call a collateral circulation develops. In other words, what happens as we get older is that this vessel starts to give off branches. And as it gives off branches, they branch amongst one another, and we get branching and branching and branching. And it joins up with branches from the other of the 4. So we're going to get all this interdigitating, this additional circulation. Now, if any one segment is blocked, all this additional circulation is available to make up the difference. Now what I've drawn in here is what a 60, 70, or 80-year-old heart would look like. The 30 or 40-year-old heart looked like that clean one, the one where there was no collateral circulation. We're getting progressively younger now in this drawing. Right? So if you lose one of these vessels, it's a major event. It's going to cause a heart attack. And in a younger, say 30 to 40, maybe up to about 50 or so, survival is less likely to happen. And then as this collateral circulation is developed, the ability to survive a blockage is much more likely to happen. Now this blockage in this area here, it only affects a small area, relatively speaking. So much more function is going to be retained. So the advantage of an older heart is all this extra circulation that allows you to survive a blockage in one vessel because of all this branching and sharing of the 4 vessels, new branches all throughout. Do you follow me on that?
Yes, sir.
Yes.
Okay, so the older you get, the more likely you are to survive a heart attack. And that doesn't seem intuitive. That doesn't always seem logical, but this is why. You may get more heart attacks as you're older, but they're often, not always, we understand there's exceptions to the rule, but often you survive them. You know, there's a good chance that an 85-year-old individual, upon their death, and you looked at their heart, might indicate that they've had 3 or 4 heart attacks, 3 or 4 clots or interruptions in blood flow that resulted in some cells dying. But they were such small areas that they didn't have a huge impact on their quality of life. So as we age, we develop this so-called collateral circulation, which gives us branches and a better chance for survival if 1 or 2 of those small branches are blocked as compared to one of the big ones in the original arrangement. So that's just something to be aware of with respect to our coronary circulation. Now, remember at the beginning, I mentioned that the cardiac muscle cells have autorhythmicity. So atrial cells they might beat at somewhere in the range of 90 beats per minute. And ventricular cardiac muscle cells might be closer to 20 beats per minute. So if we looked at our heart, and we have cells up here going at 90 beats per minute, and cells down here going at 20 beats per minute, and this is autorhythmicity, in other words, they're beating on their own accord, we've got a little bit of a problem with respect to ensuring we get one-way blood flow through those 4 chambers. Does that make sense? Do you see where we might have a problem?
Could you please repeat it
Yes.
again?
Okay. If you've got cells that are beating at 90 beats per minute at this end and 20 beats per minute at this end, we've got no coordination. And as a result, you're going to have random contractions, and the blood is definitely not going to be able to move in one direction from the atrium to the ventricle and from the ventricle through the semilunar valves, from the right side to the lungs, from the left side to the aorta. Right? So we need a system that's going to coordinate these contractions.
Yeah.
Okay? This is somewhat random, so we need coordination. And we're going to do it through what's called an intrinsic, fancy word for just meaning it's inside, conduction system. Okay, so keep in mind that if we think about our 4 chambers, we're looking at an arrangement that's something like this. We don't need these in here anymore. And our conduction system, we're going to draw the conduction system in the color yellow. So the conduction system is made up of components. Most of the cardiac muscle cells are contractile in nature, and that makes sense for what we understand about muscular tissue. It's contractile in nature. It's going to contract and cause tension. That's what muscle cells do. So the problem with cardiac muscles is they beat all by themselves. So that's not going to be terribly helpful, especially when they beat at different rates and are not really dependent on a lot of control mechanisms unless we introduce a control mechanism. So what we're going to introduce is a system of nodes and a system of conduction that we call the intrinsic conduction system. So a small percentage of cardiac muscle cells in the heart wall are described as being conductive cardiac muscle cells as opposed to contractile. The contractile ones are the ones we spent all that time talking about, with gap junctions and desmosomes and creating a functional syncytium. The conducting myocytes are kind of a combination between neurons, but are found amongst the muscular cells of the heart wall. And it's going to start with a cluster of specialized conducting muscle cells called a node. In this case, this node is located in the upper central corner of our right atrium, and it's the pace setter for the heart. This is where the pace setting is going to get started. So it's called the sinoatrial node, or the SA node. Okay, now it's nicknamed the pacemaker or the pace setter for the heart. So this is where it's all going to start. And it can start here because we're going to have innervation from the autonomic nervous system that initiates excitability in the SA node. So we know that the sympathetic division is responsible for fight or flight. So what will fight or flight do to heart rate?
Increase.
Please.
Right. Right. It's going to increase the heart rate and the force of contraction. And the parasympathetic is antagonistic to that. It's going to do just the opposite. So it'll slow down heart rate and force of contraction. So that's why it's described as the pacemaker. Now, from the pacemaker, we're going to send out electrical conduction to the left atrium and to the right atrium. Now look where this node is located. It's located superior. Where are the valves located, relatively speaking? Below that, correct? So when we contract the atrium, muscle tissue is going to start superiorly and it's going to travel inferiorly towards the open valves. Okay, so you can see that if I did this, showed you this way, the direction of contraction is from up to down, from superior to inferior. And where do we want to deliver? Inferior, through those valves. So you can see by the beginnings of this conduction system that we've already coordinated the atrial chambers to contract from superior to inferior. Remember, these cells were connected to one another through intercalated discs. When the sympathetic nervous system depolarized the cells nearest the SA node, they began the depolarization process that would then be transmitted through adjacent muscle cells in the chambers that they're most closely associated with. In this case, the atrial chambers. You follow me? Top to bottom, from the location that it began, traveling down towards the open valves. Does that make sense to you?
Yes, sir.
Okay. So what's going to happen next is that we also send signals to a second node in the pathway. So we started here, and we're going to go to a node that's located right near the junction between the atria and the ventricles. So it's very conveniently called the atrioventricular node. So that's the second location for these conducting signals. Remember, conducting was not contracting. So there's going to be a delay between the sinoatrial node and the AV node that allows the atria to contract. Now, this sinoatrial node has relatively high resistance along with the septum between the atria and the ventricle. Okay, so that atrium and ventricle septum is this one that separates the atrium from the ventricle. So as the electrical activity travels towards our SA node, there's going to be a delay because of this resistance. And there's a delay because this stuff is not conducting, it's not contractile. So the idea is we're going to get a delay between atrial contraction and ventricular contraction. We'll get even more of a delay because of where this electrical activity is going to go from here. So number 3 in our pathway is what's called the atrioventricular bundle. It's sometimes known as the bundle of His. It looks like that word should be his as opposed to hers, but it's actually a person's name who I believe was German and it was pronounced His. But it's much more accurately described now as the AV bundle because that at least tells us to look for it near the AV node. Okay, so that's a bundle of conducting, not contracting tissue that leaves the AV bundle and travels down this septum between the two ventricles. It's the AV bundle. Okay, so again, creating a delay. And look at the direction we're going. We're going towards the bottom of the heart. From the AV bundle, we're going to divide into AV branches, right and left. So a right bundle branch is going to travel down the right side, and a left bundle branch. So this means it's a bundle means there'll be several of these pathways, but the point is they're going to finish down here at the apex, and they finish on another type of tissue that's called the Purkinje fibers. And they finish on the ventricular myocytes. Now look what happens when they finish on the ventricular myocytes down here at the apex. They're going to begin to contract. And which direction are they going to travel? They're going to travel back upwards from inferior to superior. And look what we have superiorly. Our semilunar valves. So the contraction wave that's created by the delay from the AV bundle to AV branches to Purkinje fibers allows the wave of contraction to begin inferiorly and travel superiorly towards the exit point. Now, as the ventricles contract, they're going to force these valves to close and these valves to open. So the wave of contraction is going to create one-way flow towards the valve. That wave was able to be created because of the functional syncytium that connected adjacent muscle cells in the wall of the ventricle. And when you start the contraction process inferiorly, it's going to travel superiorly. In the atria, we started the contraction superiorly, and due to the functional syncytium, we created a pathway inferior, top to bottom, and down here, bottom to top. Now, if you're having a little bit of trouble following this, think about a tube of toothpaste. When you open up your tube of toothpaste, if you squeeze from the middle, there's a good possibility that toothpaste could be lost through the broken end that you could create from that pressure. But if you took the cap off and you squeezed from the bottom, the toothpaste leaves through the opening at the top. So all we've done here is taken the four chambers, looked at them as two parallel pumps, but we emptied the atrial chambers first by contracting from top to bottom where the valves were. So that's efficient. And then once we filled up the ventricle and created that electrical delay from 2, 3, 4, and 5, we now begin the contraction in the ventricle from the inferior part of the chamber and contract back towards the superior. We're squeezing from the bottom of the tube. And in the case of the atria, we're squeezing from the top, but that's because the opening is at the bottom. Does that make sense?
Yes, sir.
Okay, so I think that this is such a great example of an efficiency, a great example of a design that's meant to coordinate the random contractions of the atrial cells from the ventricle cells and coordinate them in a way that not only creates a delay between the atrium and the ventricle, which is good, so the atrium gets to squirt that last little volume of blood into the ventricle, but also create one-way contraction through those sheets of muscles that represent the chamber wall. Top down in the atria, bottom up in the ventricles. It's a great design, and it's established because we have an intrinsic conduction system that's going to coordinate the otherwise kind of random autorhythmicity of those cells. So now we're really taking advantage of autorhythmicity and of those intercalated discs to create coordinated chamber contraction towards the valves. So you can appreciate that when this ventricle contracts here in our right ventricle, when it contracts, it's going to generate pressure. And that pressure will make sure that these valves close and these valves open. So at any given time in the cardiac cycle, only one pair of valves is open and its other pair are closed. The AV valves are going to be open together and they're going to be closed together. And that's going to be dependent very simply on whether the pressure is greater in the atrium when they'll be open or in the ventricle when they'll be slammed shut. Okay, now I think I have a picture. Let's see. Slide number. Let's get this out for a minute. See if this is more or less helpful. It's small, but we can see exactly what we've been talking about here. So here's our pacemaker. And as a result of signaling the pacemaker through either the sympathetic division or the parasympathetic division, or even both, we're going to create a rate of signaling. And this rate is stronger than autorhythmicity, so it overcomes autorhythmicity. And this is what's going to initiate the atrial contraction, and it starts superior and moves inferior through the valves. Whoops, not that valve. This valve. And you can see right now, both these valves are open, and these represent the chordae tendineae that are just flopped open at this point. So we're going to move from the sinoatrial node, we called it number 1, to the atrioventricular node, the second node in our little diagram. That creates a delay between atrial contraction and ventricular contraction that's further delayed from the bundle branch, AV bundle, the bundle branches, and the Purkinje fibers. And the Purkinje fibers contract up towards the valves. There's the valve for the aorta right there, and the valve for the pulmonary trunk is what I just drew in black right there. Okay, so we're getting a contraction from bottom up in the ventricles and from top down in the atrium. And the delay is created starting at the AV node and added to by numbers 3, 4, and then finished on 5. We doing okay? We see that?
Yeah.
Okay. Now, what that looks like on an ECG, some of you maybe have seen an ECG and maybe wondered a little bit about what's going on. And an ECG just measures the electrical activity in a cardiac cycle. So the atrial contraction versus. So when we look at the base level of electricity, that's our line here. The atrium contracts, bit of a delay, and then we get ventricular contraction. And then we get our continuation on to the next cycle. So this electrical activity that we've just drawn in a typical ECG cycle, this represents a cycle. This is designated the letter P. P wave equals atrial contraction. And this is designated as the QRS complex, which is otherwise known as ventricular contraction. Okay, and then the QRS, the T wave here is when the ventricle is repolarizing, because that also requires, so it's the ventricle that's repolarizing, that is going to show up as some electrical activity as well. We can't see the atrial cells repolarizing because that would be underneath the QRS, so it's buried by the ventricular activity. The ventricular activity is much larger, so it covers it up. So QRS represents the ventricular contraction group, the points of beginning and end, and the peak. So this is what you see when you see somebody's ECG repeating itself. This represents a cardiac cycle. And the cardiac cycle are the events that are going on in the heart chambers that push the blood from one segment to the next one. So our cardiac cycle is coordinated by the intrinsic conduction system that also creates changes in pressure. So when we look at a cardiac cycle, because it's a cycle, we're really just looking at the changes that happen in a chamber with each heartbeat. So if it's 60 beats per minute, these changes that we're about to describe are going to happen 60 times a minute. If they have to happen 100 times a minute, these same changes in this same sequence is going to happen 100 times per minute. So we can explain why it happens because of this intrinsic conduction system that coordinates muscular contraction in the chamber walls. Remember, those chamber walls are going to act like a sheet, and there's going to be a slight delay, so more like a wave of contraction. So our cardiac cycle has got relaxation phases in it and contraction phases in it. And in the cardiovascular system, when the walls of the heart are in a relaxed state, the heart is considered to be in diastole. Relaxed myocardium. And systole is a contracted myocardium. Now, from a functional point of view, in other words, from the pump point of view, we're especially interested in what's going on in the ventricles. So if you don't designate specifically where the relaxation or the contraction is occurring, there's an assumption from a functional point of view that you're talking about the ventricle. If you want to talk about atrial contraction, you need to make sure that you put the word atrium in front of the word systole, and vice versa if you want to talk about diastole. So when we talk about any kind of cycle, in theory, you can start anywhere you want. You can start in one location, as long as in the cycle you return back to your starting point. So by convenience and by convention, we're going to start our cardiac cycle in what we call mid to late diastole, and you'll see why. So we're going to start in mid to late Now, in this case, we didn't specify whether we're talking about atrium or ventricle. So in reality, we're talking about the ventricle. But this is the point, mid to late diastole. And the reason we start here is that all chambers are relaxed. So all chambers in other words, they're all in diastole. And so if all the muscle walls are relaxed, pressure is low in all the chambers as well. And blood flow freely from the atria to the ventricles, because blood is being returned to the heart through the vena cava and through the pulmonary veins back to their respective chambers. So it's going to flow into the chamber. The AV valves are open, so they're going to flow freely from the atria to the ventricles. The blood comes back from the systemic circulation and or the pulmonary circulation to their respective atria. The AV valves are open, the blood flows freely from the atrium into the ventricle. But the semilunar valves are closed. Remember, we said at any given time, if one set is open, the other set will be closed. So that creates a chamber, the ventricle, where the blood has nowhere to go except to fill up. So in this phase, the ventricles will be filling. And just as we approach the end of this phase, late diastole, then the atria contract. And a final extra little volume of blood will be pushed into the ventricle. Okay, so that means we're going to move to the next phase, which is systolic for the ventricle. So our next phase will be ventricular. Okay, so we're going in a progression. So ventricular systole means that the ventricles are contracting. So they now have a volume of blood in them, and they're going to begin to contract. So this would be our second step. Our first step was at mid to late diastole. So the ventricles contract, and that's going to occur just after the atria did. Remember that delay was created by that second node and the passage through the bundle, and then the bundle branches to the Purkinje fibers. So when the ventricles contract, that contraction causes a big increase in pressure within the ventricle. And that forces the AV valves to snap shut. And that's otherwise known as our first heart sound. Some of you maybe have used a stethoscope in the past, and maybe you've heard that the heart sounds are described as a lub-dub, lub-dub, lub-dub. And this is the lub. This is the first of the heart sounds. And it's caused when the AV valve snapped shut, and that was caused because the ventricle contracted and caused a big change in pressure. Remember, when the pressure in the atrium is larger than in the ventricle, the valves are open. Now they're shut. So we force the AV valves to open. Sorry, we force the AV valves to close, and that's going to add even more pressure inside the ventricle because, at least briefly, there's nowhere for the blood to go, and the walls are continuing to contract. So next, the semilunar valves open. So that causes the semilunar valves to open, On the right side, the blood will be delivered into the pulmonary trunk to ultimately be delivered to the lungs. And on the left side, the aorta to be distributed throughout the systemic circulation. And then the larger arteries, blah, blah, blah. We get that distribution going on there. And during this time, the atria are relaxed and filling with blood. So now when the ventricles relax, which is our third step, in our third step, ventricles relax. So we're going to call that early diastole. Remember, we started in mid diastole. So the ventricles relaxed, and that drop in pressure causes the semilunar valves to close. So again, briefly, all 4 valves now are closed, which is not allowing blood in, and the blood that's already left, so the pressure will continue to drop. Okay, but when those valves close, that represents the second heart sound. So it's the valves closing that are responsible for the heart sounds we hear through a stethoscope. So because of the semilunar valves to close, so there's a drop in pressure because all of a sudden the contraction stopped. That drop in pressure causes semilunar valves to close. Now there's nowhere for blood to go and no reason for the blood to go anywhere. So the pressure continues to drop. And once the pressure is less in the ventricle, the AV valves open and blood flows into the ventricle because it's been collected by the atria all through this time. So we can look at this. There's another summary of this here. I believe not there. Here. Okay. So if we look at our ventricular systole here, so let's start here. Ventricular contraction pushes the AV valves closed. Okay. And in the second phase, semilunar valves will open and the blood gets ejected. Okay. Semilunar valves are going to close once contraction stops, early diastole. And in ventricular diastole, mid to late, chambers are relaxed, blood fills, AV valves are open, semilunar valves are closed. Then the atrial contracts just at the end of this stage. So we get a contraction, which is our P wave. QRS is our ventricle, repolarization, so on and so on. Ventricular systole, ventricular diastole, and this is a continuation of ventricular diastole. But at the end of ventricular diastole, we get an atrial contraction that initiates that extra spurt of blood that's going to be delivered to the ventricle wall, to the ventricle chamber, I should say. Okay, so it's a sequence of events that are driven by contraction and relaxation. And the contraction and relaxation is sequenced by the intrinsic conduction system in our cardiovascular system. Okay, how are we doing with this? Are we okay? We seeing how this develops?
Yeah.
Okay, so it's just a question of understanding, like physiology usually is. It's a question of recognizing that when one step's finished, another step can begin. And you can't get the first step, or that second step, I should say, unless we had the first step before it. Okay, now, what's the purpose of all of this? What is the purpose of having valves open and close and chambers fill and empty and all this kind of thing? From a physiological point of view, the purpose is to establish what's called the cardiac output. So cardiac output is literally what it sounds like. It is the output of the heart by measured as a blood volume. So the good news is we can summarize cardiac output as being the amount of blood ejected by each ventricle per minute. So this is a measurable volume of blood that is measurable and determines the health and effectiveness of the cardiac function. So what's even better, although not everybody agrees with me, is that we can summarize cardiac output with an equation. Some people get excited about equations in a bad way, but this is a way to summarize some of the things that we've been talking about and sort of simplify it by putting it all together in a pretty straightforward equation. So the equation looks like this. Cardiac output is a function of heart rate times something we call stroke volume. Now, heart rate, we know, is going to be controlled by the autonomic nervous system. Cardiac output is a volume of blood ejected by the ventricle per minute. Heart rate is measured per minute. That's where the time comes from. Stroke volume is the volume of blood ejected by the ventricle each beat, each cycle, in other words. So as blood is being returned to the heart, we're creating the volume of blood that is going to become stroke volume. So stroke volume is a function of the volume of blood that's going to be ejected. So if we looked at 1/2 of the heart, so let's say we're going to look at the left heart, okay, and here's the atrium, and blood is going to fill up on the left side of the heart. So I'm just going to, I'll put the right side in here just so we have a comparison. The idea is you could measure the right-sided stroke volume, and in a healthy heart, it should be pretty close to the same, but it's easier for us to look at what's happening on the left side, and the stroke volume becomes the volume of blood being ejected to match the demand of blood flow in the systemic circulation. So what we have is a volume of blood right here. Whoops, wrong color. So that volume of blood that we see there is described as our end diastolic volume. Okay, so and we're going to keep the numbers nice and simple. We're going to say that the end diastolic volume in our example is 100 milliliters. Okay, then we're going to go through ventricular systole, contraction, and we're going to eject a volume of blood. when we eject that volume of blood, we're going to have a volume of blood after ejection that's maybe down in that vicinity there. So this would be described as the end systolic volume. Okay, now some people are going to say, well, why wouldn't you just eject everything? If it's so important to deliver oxygen to all the blood, all the cells in the body, why not just eject the whole 100 milliliters? Does anybody see a problem with doing that? Well, you can start your answer by saying, yes, I see a problem, or no, I don't see a problem. So my question, I'll repeat the question, is why doesn't the ventricle eject the entire volume? So in our example here, we're using 100 milliliters. Let's eject the whole 100 milliliters with each beat. Do you think that might lead us to a problem?
Yes, I guess.
Okay, that's fair. So the short answer, the part I'll agree with you on, is yes, it's going to lead us to a potential problem. So remember, we like to think of these systems in terms of efficiencies. So we did a good example. We've done a couple of examples of efficiencies already. The fact that the cardiac muscle wall is a functional syncytium, that's an efficiency. The fact that the wall of the ventricle contracts from the bottom up, that's a type of efficiency. The atrium from the top down, that's a type of efficiency. The reason that we don't empty the whole 100 milliliters out with each beat is that means we'd have to fill up the whole 100 milliliters with the next cycle. And the chances are that if you emptied the chamber completely, the chamber might want to collapse. That's not very efficient. So we leave a bit of volume in there that starts us on our next cycle. But the point is, we have created an ejection fraction. So now we have or sometimes it's often referred to in a percentage, but it's based on, so in our example that we're going to use here, we're going to leave behind about 30 milliliters. So our ejection fraction was 70 milliliters. And if we describe that ejection fraction in a percentage, it means that our percentage was 70%. And we want a healthy ejection fraction to be somewhere in that 60 to 70% range. So I used some simple numbers to get us to 70%, just so we could see how it happens. Okay, so a good healthy stroke volume has an ejection fraction of around 70%. And if you drop very much below 50%, it's indicating that the heart's not doing its job. Okay, if we get somewhere between 50 and 60%, it might not be noticeable right away, but much less than 50, and it becomes noticeable, and you're not meeting minimum demands anymore. So if we take this formula up here and we say cardiac output is a function of heart rate, and we say that a typical heart rate is about 75 beats per minute, and now we put in our ejection fraction, our stroke volume, 70 milliliters with each beat, we're going to get a number. And it just so happens that I've got this number memorized. These are not numbers for you to memorize. The equation is worth memorizing. And if I was going to ask you to figure something out, I would put the numbers in for you. But this is the equation. So this would be 5,250 milliliters per minute. Now, does anybody know what an average blood volume is in a typical human? Total blood volume in liters. Anybody have any idea?
Five.
5? Well,
5,000?
Pardon me?
5,000 milliliters?
Okay, you know what I'm going to say to these answers.
Yeah, approximately 5 liters.
Okay, well, first you said 6.
No, 5,000 milliliters.
Okay, but first you said 6,000 milliliters. So which
No.
is it?
No.
Oh, you didn't? I misheard
I
that
didn't
somehow?
say 6.
Okay, but what's the point I'm making here? Somebody tell me what the point is that I'm making. Somebody said 5. I thought somebody
What you're
said 6.
making is you want a range. I think
I do. I want a range. That's absolutely what I'm looking for. So the one person said approximately. Okay, that's acceptable. But what I want to see is we understand there's a range. So in a typical male, where most of the research has been done, a typical blood volume is somewhere between 5 and 6 liters. Here we see that cardiac output is pumping, in our example here, a little more than 5 liters per minute, which means that cardiac output every minute is pushing through the total volume of blood through the body. Wow, that's a lot of push. That's a lot of volume every minute that we're at rest. And would you think cardiac output would go up or down if we exercised?
Up.
Yeah, and you can look at this equation and very quickly tell me why. Because what else goes up during exercise?
Heart rate.
Heartbeat.
Heart rate, sure. So under fight or flight conditions, cardiac output goes up. What does cardiac output represent? Oxygen and nutrient delivery to all the cells of the body. What do they need during fight or flight? They need more, because we need to make more ATP to fight or flight. So this equation explains why cardiac output would go up during exercise or any other type of stress, right? And we can reverse it and say, well, it'll go down. If we slow down the heart rate, we can reduce cardiac output. If we could increase the ejection fraction or increase the stroke volume, cardiac output goes up. But in the example we've used, which is fairly typical at rest, what we're looking at is that every single minute of every single day, the heart pumps the equivalent of the total blood volume throughout the body. And that's kind of amazing when you think about it. Every minute, 5 to 6 liters of blood is going to be pushed through the entire circulation and repeated every minute of every day that you're alive, to varying degrees.
Sorry, so that means during the exercise, the ejection fraction is more than 70%, right?
So during exercise, the ejection fraction is
more than 70%.
It could be. It could
Okay.
be. But the idea is that it's probably going to be maintained somewhere between 60 to 70%, but the other numbers are going to change.
Mhm.
So because your overall circulation would go up, your end diastolic volume at 100 would probably go higher, say to 120. So your ejection fraction would still probably be 70%, but it would be a higher number than 70 milliliters because we started at 120, right? And then so 70% of 120, let's say it's going to be 80 milliliters. So stroke volume goes up, not so much, excuse me, not so much the ejection fraction, but the overall volume will definitely go up. with exercise. Does that make sense? You okay with that?
Yes, sir.
Yeah, thank you.
Okay, so I just want to take a few more minutes to talk about this stroke volume. What makes stroke volume better or worse? Okay, so this is sort of an important thing with respect to our discussion on cardiac function. So we'll put our equation up here. Cardiac output is a function of heart rate times stroke volume. Stroke volume is the volume of blood ejected each beat, or each cycle, if you like. So stroke volume is dependent on 4 major factors. Stroke volume is going to be dependent on something we call venous return. And that's the amount of blood returned to the heart in a cycle, right? Or in other words, in a beat. So if we don't start with a volume of blood, we're not going to have a strong stroke volume, okay? So venous return is defined as the amount of blood returned to the heart in a cycle. The amount of blood that's going to be returned to the heart and then filled in the ventricle is going to be called preload. So preload is the stretch on the ventricular wall. So basically, when we talk about the stretch on the ventricular wall, we go back to that balloon. If we put a stronger stretch on the balloon wall, we get a more crazy release when it flies all over the room. So within reason, more stretch on the ventricular wall is going to give us a more forceful contraction. Okay, now if that doesn't seem quite right, try to remember this. If you remember what the sarcomere looked like, we had thick and thin filaments. And remember, the thick and thin filaments, they were kind of connected, created Z lines that looked a bit like this, right? And that represented our sarcomere. That line should not go that direction. Oh yeah, I guess it does go that direction, right? So this represents our sarcomere. Everybody recognize that okay?
Yeah.
Okay, so the overlap between the thick and thin filaments is what determines how much force of contraction we can have. So if we put a preload on this, our thick filaments, they can't change. So here's our thick filaments, same size, in the same location. But because we put a stretch on the muscle tissue, what we're going to do is see us go out here. And we got a so now our sarcomere is lengthened. So we've got a longer sarcomere up here than we do here, which means that our space in here is bigger. What that means is, remember when we contracted, as long as we had ATP and as long as we had calcium, we could continue to contract. And that meant that the myosin heads would break off and reattach and then re-ratchet, break off, reattach, re-ratchet, so on and so on and so on. And our sarcomere would continually get smaller, but there's a limit. It can only go until the thin filaments meet in the middle. Then there's nowhere else to go. So if we have a bigger middle, we can have a more forceful contraction. So up here, this was our middle. Down here, this is our middle. So down here, with a preload stretch on the ventricular wall, we can have a more forceful contraction. Does that make sense?
Yeah.
Okay. Now, here's my favorite question of the day. Does anybody see a potential problem with preload?
It can overstretch.
And if we overstretched, what we're talking about then is we would lose the integrity between the thick and the thin filaments. They would no longer be able to contact each other to create the cross bridge because they're overstretched now. So you're absolutely right. Preload is very good within limits. And preload is directly related to venous return. So if we returned too much venous blood, we could create too much preload. But that's pretty rare. There are some, you'll learn a little bit about that in pathology. But so it can happen. But within reason, that's the advantage of a preload. Putting a stretch is going to give us a more forceful contraction. And it's explained if we remember generally what our sarcomere looks like. So another factor that affects stroke volume is contractility. These are the properties affecting contraction. So availability of sodium, of calcium, of the autonomic nervous system working properly, acid-base balance, the availability of oxygen, and so on. So these things that make a muscle cell contract efficiently is what we're talking about when we talk about contractility. So now, watch this. This is sometimes tricky for people. If I say that I want to be able to increase stroke volume, so now I've put a value on stroke volume. I want to make stroke volume better. To make stroke volume better, the shorter answer is we want to improve venous return, improve preload, and improve contractility. The reason I bring that up is there's a fourth factor, and the fourth factor is not called preload, it's called afterload. And afterload is the resistance on the other side of the semilunar valve. Okay, so afterload is a problem because if resistance is high on the other side of the semilunar valve, okay, so what that means is if we're looking at our left ventricle, remember the left ventricle is going to eject through this semilunar valve, which then enters into the aorta, right? So if the pressure is high in the aorta, that's afterload. And that means this ventricle has to contract even more against the afterload. So if we're having a discussion about factors that improve stroke volume, that make stroke volume go up, then it's about improving 1, 2, and 3 and decreasing number 4. So think of afterload like blood pressure, hypertension. If the pressure in the aorta is very, very high, maybe because somebody has high blood pressure, that's harder on the heart. That reduces efficiency. So in the interest of maximizing or improving stroke volume, which in turn improves cardiac output, we would like a good, healthy venous return. We would like a good, healthy preload, the factors that improve contractility to be available, and we would like to minimize resistance on the other side of the semilunar valve. We would like to minimize afterload. Okay? Does that look a little bit like a multiple choice question? I'll tell you one thing, it also looks a little bit like a short answer type of question.
Yeah.
So something to think about, right? So these are factors that are going to have an effect on cardiac function. And we can explain a lot of those factors by understanding that equation. Okay, so I think that's enough for today. Next week, after our test, we will continue with the cardiovascular system in the classroom. And then, as I mentioned, we will continue with blood vessels from once we get the heart finished up, and then look at the immune system after that.